Hello everyone, welcome back to one more video session. I'm continuing with the same chapter, that is sexual reproduction in flowering plants, and in the previous session I have explained about the endosperm, that is the first step of post-fertilization event. Now I'm going to talk about the second step, that is the formation of the embryo, so which is very important for NEET. First we'll write embryo. Okay, so this is the embryo. The process of formation of the embryo is called as embryogeny from the diploid zygote, that is embryogeny. It is also called as embryogenesis. Just remember embryogeny. Okay, here I am going to draw the diagram of the embryo sac because this is the site where formation of the endosperm as well as formation of the embryo takes place, you know it. Okay, so here I am not going to draw other cells because they have degenerated already, because endosperm is formed completely. Now the zygote is undergoing some cellular division to form the embryo. Only two things are remaining, that is endosperm and the zygote or embryo. Only two things are remaining. So here we'll write this is the micropylar region and this is the chalazal end or region. I am going to draw the diagram of zygote. So this is the zygote. The ploidy is 2N and the zygote is surrounded by millions of cells, and these cells are nothing but endosperm. Correct. So we have this is endosperm and this is zygote. So what is the ploidy of endosperm? 3N. Remember this. The ploidy of zygote is 2N. Correct. But I am going to explain about embryogenesis. So I need only zygote now. Okay, and remember why this endosperm is surrounding the zygote, because it has to provide nutrition to the developing embryo. Correct? Okay, so now I am going to draw the same diagram. Here I will draw inverted diagram of the embryo sac. Inverted in the sense the chalaza will come this direction, whereas the micropyle will go in this direction. Okay, so micropyle is here, chalaza is here. It is convenient for me to explain the concept. That's it. Okay, fine. Now we'll draw the zygote. Zygote is here, right? So zygote is here towards the micropylar region. Correct. Okay, I will not draw the endosperm. That is not required. Okay. Okay, so here I will just draw the zygote outside. So this is the zygote now. We have zygote here. What is the first step of formation of the embryo from the diploid zygote? That is the cell division. What kind of cell division occurs within the zygote? The first type of cell division is the transverse. I'll write here. What is the meaning of transverse? Horizontal. Correct. Horizontal. Transverse cell division occurs. That means this zygote undergoes transverse cell division like this. This is transverse, horizontal cut. And what happens? Two cells are produced. That means one zygote cell now becomes two cells because of the transverse cell division. As a result, two cells are produced. Now, this cell is closer to the micropylar region. Correct? For example, okay. As a result of cell division, we have two cells. One cell is near to the micropyle. The other cell is towards the chalaza. Correct? Correct. So can I write this is micropyle, this is chalaza? Correct. Okay. Now I use color. I use pink color for the micropylar sided cell and I use blue color for the chalazal sided cell. Okay. So that you can understand this better. Okay. Now the cell which is closer to the micropyle, which is actually called as the basal cell. Basal cell, the cell which is the product of the zygotic cell division, and this one, the blue color cell, which is closer to the or towards the chalazal side, which is called as terminal cell. Remember this. Basal cell and terminal cells are produced from one zygote. Okay. Now what happens? In the basal cell, you could see it is also called as, it can be denoted as CB. It's only noted for NEET examination. CB means the basal cell, and this one can be written as CA. So we have CB and CA, that is basal cell, terminal cell. Correct? Okay. Now let us see what happens now. Now the basal cell, I am going to just continue this. The cell further, the two cells further undergoes mitosis because it has to increase the number of cells to form embryo. Correct? Okay. So it will undergo mitosis. I'll just write M here and I'll draw the pink cell now. That is the basal cell. The basal cell again produces two cells. It undergoes cell division and produces two cells. Okay, whereas this one, the terminal cell or CA, again undergoes cell division and produces two more cells. Okay, but remember that the type of cell division is slightly different. Okay, the mode of cell division is slightly different, and in this case you could see this is horizontal, that is transverse cell division, and in this case it is longitudinal or vertical cell division, correct? And how it looks? It looks like inverted T, correct? It looks like this. This is the first embryo. Okay, so inverted T-shaped, and two cells are the basal cells, and these two blue cells are terminal cells or CA, okay, or chalazal sided cells. Okay, so this is because of the transverse cell division, and this is because of the longitudinal cell division. Just remember it, and as a result you see this inverted T-shaped embryo, and this embryo contains 4 cells. 1, 2, 3, 4, correct? 4 cells are there. I'll just write 4 cells, and this embryo is called as the pro-embryo. Remember this is the first stage of embryo formation, the pro-embryo, the first embryo. And again the process will not stop here, it continues further. Let us see how. I'm just going to draw the arrow mark here. Again the mitosis continues. Okay, let us see. Now what happens to the pink cells? I'm talking about the pink cell. Pink cell again undergoes cell division, producing 4 cells, correct? 2 cells produce 4 cells. Again this one, the blue color ones, produces 4 cells, correct? Okay, how exactly, how many cells are there now? Total 8 cells are there. Okay, so again the cell division continues further, producing like this. How many cells are there? 3, 4, 5, 6, 7, and the last cell is bit bigger in size. Okay, I'll tell you the name of that. Okay, and here the blue color cells continue to divide further, and as a result, total how many cells are produced here? Here 8 and here 8. Total number of cells this in this embryo 16, correct? So let me write 16 here. Total 16 cells, and the shape, see here, it looks like a globe. Correct, it looks like a globe, and hence this embryo is called as globular embryo. This embryo is called as globular embryo. Now, this particular globular embryo is very important for NEET. You have to remember what is the structure now. See, this is actually the result of continuous cell division within the basal cell and produced 8 cells, and this is actually 8 more cells from the terminal cells. Okay, so this here, let me tell you, this is the first cell of the basal cell. Okay, and this is called as hypophysis. The hypophysis cell is very close to this globular shaped blue color cells, correct, or the terminal cells, very close. In the future it's going to form the radicle. So what is radicle? Radicle means it is the tissue which produces the root tip. It's going to produce the root tip. Okay, radicle. And then you could see a chain of cells. These chain of cells are called as suspensor. You may get this question, like diagrammatic question, so you have to label. So this is the suspensor. What is the function of suspensor in the embryo? It gives physical support to the embryo. You got it? Okay. The last cell is the largest cell. It is the terminal cell, which is vesicular cell. Vesicular cell. Okay. So this is the vesicular cell. Fine. Okay. Now this is the globular embryo. Again the cell division continues further. Let me just put one more arrow mark. Okay. Again the mitosis continues, and as a result what you see here is the pink. Okay. The number of cells increases, and again here also the mass increases because of the cell division, and as a result you see a shape like this. Just shade it. Anyhow, all these things are nothing but covered or filled with the cells. Okay, how does it look? This looks like a heart, correct? Hence this embryo is called as heart shaped embryo. This is the third stage, heart shaped embryo. Remember it, heart shaped embryo. And now in this particular embryo the differentiation of the cells takes place. Due to the differentiation the embryo becomes mature now. Okay, final stage of the embryogenesis is formation of the mature embryo here. So what happens? You could see a mature embryo. This is the mature embryo. Okay, That means actually the direction of the growth is towards the chalazal side. Correct. The embryo formation is towards the chalazal side, and this is the mature embryo, and this is called as mature embryo. Mature embryo. Okay. Hope you understood this concept. Simple. First stage of embryo formation is proembryo, then comes globular embryo. Remember, this is very important. Many labelings are there, that's what. And then actually heart-shaped embryo, and finally the mature embryo. So this is the dicot embryo. I told you in the beginning itself. Okay. So here you can you tell me what is this? These two are the cotyledons. Okay. I'm going to explain that. I'll give you the detailed explanation about dicot embryo as well as monocot embryo later. Just remember this. This is actually the general concept how exactly embryo is produced from a single celled zygote. Okay, hope you understood this concept.